All right, so in this video, we're going to be talking about genetic engineering and biotechnology. There used to be a whole lot more of this in AP biology, but they've taken quite a bit of it out, which is fine with me. And so basically, genetic engineering that's not the right place. Here it is. Genetic engineering is the idea of the human genome or any genome being influenced from the outside, right, on purpose. You see this person inserting a section of DNA into the model. So yeah, there's that kind of goofy looking, but it gets the point across that this is what's going on. We're going to talk about three specific techniques in order to for genetic engineering. We're going to talk about gel electrophoresis. We're going to talk about PCR or polymerase chain reaction, and then we're going to talk about DNA sequencing as well. We're also mentioning some bacterial transformation that we've talked about already. So gel electrophoresis is a technique that separates molecules of DNA according to their size and their charge. DNA is inherently negatively charged because of all the phosphates. All those phosphates have a negative charge, and so the DNA is a highly negative molecule. So if you put DNA in an area of a negative charge and you put a positive charge on the other side of that area, that DNA is going to be attracted to that positive charge. And so what happens with gel electrophoresis is they take a section of DNA. Let's say they take my DNA, all right, and then they apply a particular sort of chemical to it called a restriction enzyme. And the restriction enzyme is going to go through, and every time it sees a particular sequence of DNA, like let's say every time it sees AACAA or something like that, I just made that up, it's going to cut that DNA. And so you're going to have really long strips of DNA and really short strips of DNA all in this fluid, right? And then you inject that fluid into these little wells here of this gel. So this gel is not solid yet. And so the DNA is able to travel through it, and it's going to want to travel to the positive end. And it's going to travel through this gel. And so the idea here is that the really big pieces are going to get bogged down, right? They're not going to travel nearly as far, but the really small pieces are going to travel further. And what would this be used for? Well, if my DNA is different than yours, right? And so let's say that they find some DNA on a crime scene, and they're trying to figure out who is that, whose mystery DNA is that. Well, if they have some suspects, they can take all of their DNA, apply the same restriction enzyme to it, and because we all have different DNA, it's going to look different, right? It's going to create something that looks like this. This over here is like the standard. This is just kind of like the ruler, and this is going to show that particular person's DNA. Well, if they take the crime scene DNA and they match it to my DNA, and it looks just like this, no one else's is going to look like mine. All right, it's going to be very specific, and they can say, "Yeah, this is the right person." In that case, so it can be used to identify people specifically is one of its uses. We're actually going to do a gel electrophoresis lab in this class. Polymerase chain reaction. This was created in the early 80s, I believe. Oftentimes you'll just see it called PCR for short. And what PCR does is it takes a short section of DNA and makes lots and lots of copies of it. So one of the problems in early DNA research is having multiple copies of a gene in order to work with and do experimentation on. And so this particular uh, mechanism allows for the creation of lots of copies of that gene, which allows for infinite testing in that way. So how does it work? They heat up the DNA in order to get it to denature, and they introduce things like primase and DNA polymerase to it in order to replicate that short strand of DNA. And so now you have two pieces. Well, they do the same thing again, and now you have four, and they do the same thing again, and now you have eight, and you get the idea. And you just keep going, and you have as many copies of that gene as you want. One interesting thing about this is the polymerase they have to use has to be able to withstand high heat, and so they actually took the polymerase out of bacteria that are found in hot springs. I just thought that was fascinating. Moving on. All right, bacterial transformation. We actually use bacterial transformation. It can just happen, but we actually do it on purpose in order to create bacteria that have special tricks that they do. Here's some bacteria that have been introduced with a gene that causes them to create phosphorescence or a glow in the dark. This is the lab we're going to do in here, or at least we're going to try, see if we can get it to work. Um, well, what if you created a bacteria that can produce insulin, for instance? You have an insulin. You teach it how to make the insulin protein. Well, then you can manufacture insulin, which is exactly what they did. Um, so that's you create medicines. You can create any kind of protein doing this. You can teach bacteria to eat oil, which is what they've done to clean up oil spills. So you get the idea. This can be used using bacteria to do our bidding. Sounds kind of bad, but they're just bacteria. And then lastly is this process of DNA sequencing. This is less about engineering, more about just biotechnology. There's no changing of the DNA, but being able to take an organism's DNA and sequence it, meaning to basically figure out what the code is going to be helpful, because then you can pair organisms to one another and you can see change over time. And whereas in the past, if you found two mice out in the field and you wondered if they were related, you would just kind of look at their phenotypic characteristics and make conclusions there. But now you can just take samples of their DNA and sequence that DNA, and you can find whether or not they're truly related in that sense. And so this is advanced, highly advanced, the field of evolutionary biology and understanding and categorizing species. And it's also used for medicine, can be used in forensics, can be used in lots of applications. Essentially, what they do is they attach like a fluorescent dye to um, each specific base. So the C's are blue, the T's are red. You get the idea. And they sequence it, and you have something that looks like this. And then a computer goes around and reads them and tells you what they are. And computer has to do it because there's 6 billion of them. It would take us years, take a room full of people years to do it. And so they just figured out how to use computers to do it. So yeah, DNA sequencing, pretty important as well as far as biotech is concerned. But that's all we're going to talk about with biotech. There's a lot more that we could talk about, but these are the ones that AP requires. And so that actually brings this whole unit of genetics to a close.